Okay, hi everyone. Welcome back. So today is the final lecture that I'll be giving, and then I'll pass it over to you to present your final projects. So let me remind you a few things about how it's going to work. So next week, there will not be a lesson. We'll give you time to prepare, think, and practice. More on that in a minute. I know most of you have already sent us, your suggested subjects. So a few things about that. We'll try to get back to you soon, especially in the coming day, especially for those who are supposed to present first in 2 weeks. I would say that, if you decide to change your mind along the way, no problem. Just let us know, because maybe when you see others, or maybe when you practice on that, you find actually you want to frame the question differently, or actually jump to a completely different subject. That's also completely fine. Okay, so don't feel too attached to that.
Sorry.
A few other things. So just in terms of the timeline, because we found, so we've been practicing that for several years, and we find that the more you prepare for it, the better it is both for you and also for all the listeners. It's also a pretty good practice, in life in general. So the idea is that a week before you're presenting, by that time, you're already supposed to sit with a mentor of your choice. Somebody that you could do the real thing. You can come here. These whiteboards are usually available, or you can choose the whiteboard of your choice and do the whole thing within the time frame that's allocated. And what you need to do a week before is to send us what you took from talking with your mentor, with practicing. Something that it could be one thing, several things. That you can share with us on the email of the course. Okay, that's a week before. On the day of the lecture, you'll see we have a whole way protocol how to do it efficiently and nicely. But all those who are not presenting, you're supposed to be here. Listen to your friends, learn from it, experience, and also choose at least one person from the people presenting to whom you'll be sending feedback. It could be feedback on what you heard in their presentation, and also to ask for the vignette. You remember we have these vignettes that we're writing, so you'll give them feedback and ask for the vignettes, which they will send you the draft. You look at it and you can send them back feedback on the vignette. Okay, so that's something that you also need to do. That would also help you prepare your vignettes to write them better. The final project in the form of these few pages vignette is expected 2 weeks following your presentation to the class. Okay, protocol is more or less clear. I know it's a lot to absorb, but you're bright students, everything's fine. I forgot something? Okay, good. A few more comments about that. There's a strong constraint by the Weizmann administration that the total average grade of the course could not be higher than 90. So as you can understand, that's pretty limiting, because all of you did so far you did very well on your exercises, and I'm sure that with everything that we have ready, you give good presentations. Still, anyone that gets a 99, there's somebody else that needs to get an 81 or on average. So bear with me, and the important thing is that you're experiencing. Nobody cares too much about the final grade that you come from Weizmann, ensuring that you get your master's or PhD and continue in life with the tools you've taken. But I want to apologize in advance that even if you feel that if I will feel that everybody deserves here a 98, I cannot give you all a 98. One last thing, as you know, in all these lectures have been filmed, and we've put them so that they'll be available either for the students and for others through YouTube. What we're suggesting is that also for the final project, we'll be filming them, and then we'll be cutting it in parts so that each one could get, if they want, their own part, their 5 minutes of glory. And then if they want, we could also load it to the thing that we have of the course. Now everything here is up to you. We're not forcing you in any way. If you don't want to be filmed, you won't be filmed. If you want, after you've been filmed, that it would only come to you and not go anywhere else, that's also completely fine. Feel free about it, no pressure, it's not related to anything else, to how well you did, etc. we thought it might actually be a special opportunity for you to observe yourself and have the ability to see how, it's part of the communication skills and all sorts of things like that. Could also be fun. You can share it with the family, do whatever you want. Okay, so be aware, we have this, and you'll decide if you want or not. If you want to talk with us about that, you can also either email us or talk with Lior or Ron, etc. Questions or comments about that? Good. So let's get to business. So let's start with today is all about evolution as a very interesting tool that made all of us. And let's start with talking about at the heart of it sits our genomes. So let's start with a question. Comparing the genomes. Let's take two key organisms. What would they be? Okay, so everybody know the central organism E. coli, and the other central organism? Us. Yeah, so pretty fixated about us. So let's compare it in terms of genome length and in terms of gene content. So let's have it 5-fold. 50-fold, 500-fold, or 5,000-fold, and same here, 5-fold, 50-fold, 500-fold, 5,000-fold. Is the question clear? So I'm asking if I take these genomes and I compare them in terms of their size and their content in terms of genes. Okay, so you know the drill. Let's vote. Let's start with the genome length. Who's voting for A? 5-fold. 1 person. 50-fold. 4 people. Okay, let's start. I see nobody. I'll write it down so we could follow. Okay, so again, who's voting for 5-fold? I have 1. 50-fold. 5 people. 500-fold. Half. 5,000-fold. The other third. Okay. Somebody could then infer how many people were in the class. Okay. In terms of gene content, who's voting for
base pairs?
what?
In base pairs?
Let's say in base pairs.
No, the gene content.
Gene content is in terms of number of genes. Thanks for asking. So this is kind of like base pair. And this is for gene content, number of genes. So who's voting for 5-fold? 10 people.
Compared to so in one you're comparing the E. coli to the humans, and the other you're comparing humans to E. coli, right? How does this work exactly?
No, let's say the ratio between Homo sapiens and E. coli is 5-fold, however, and between number of genes here and number of genes here.
And number of genes. Okay.
Okay. What do we have? 50-fold. Okay, about half. 500-fold. Few. And 5,000-fold. Another few. Okay, so we have a good coverage. That was half, few, and few. Okay, so what anybody carries in their mind something about genome length?
It's 5 mega for E. coli, and for us it's 3 giga.
Okay, so E. coli about 5 mega bases. Okay, you remember mega 5 million bases, and Homo sapiens 3 giga. So we are 3 billion and our E. coli are 5 million. So indeed that brings us, well done for that half, about 500-fold. And actually, based on this, when people, the human genome has been sequenced not so long ago, around the year 2000. And just before they finished sequencing it, they knew the length already, but they didn't know yet what would be the number of genes. So there was actually a contest of who would be guessing the number of genes most accurately. And it was pretty interesting because in the end, when they came up, it was much lower than the vast majority of expectations. So they did kind of like what we did here in class, but with people who are experts, and it turned out to be quite off. So let's talk about that for a minute. What's the number of genes in E. coli?
4,000?
Yeah, so it's somewhere between 4,000 and 5,000. It's actually pretty elegant that you see the ratio is about 1,000 to 1, meaning for every kilobase, every 1,000 bases, you have about 1 gene. And that's actually true for most bacteria. And if you think about what's the length of a gene, like what's the length of a protein that's being coded?
300?
200
amino acids?
Yeah, so something like 300 amino acids.
Is about 1,000.
300 amino acids, and you need 3 bases to code for every amino acid. So you need about 900 bases, which is not very far from the 1,000. So it means that the bacterial genomes are very compact. Okay, they don't leave a lot of space. There is some, promoters and other stuff, but not a lot. It's pretty dense with genes. Okay, what about Homo sapiens? How many genes do we have?
20,000.
20,000.
Right, so that's about 20,000.
The protein products are different.
And so you can see the ratio between this and that to a reasonable approximation. It's about 5-fold. Yeah. Okay, which shows, I think I did it right when I was, asking you. It's interesting that even we're way past the year 2000, so we know the gene content very well. We still have within us, like the answers that you gave are so like reminiscent of what people were guessing before there was the knowledge of the genome content. So it shows also how we're driven by our gut feelings or by intuitions, and it's hard for us to update our software in the brain, even though there's like new information coming up. So it's really well established now that the ratio between this and that is actually not very large. It's just against our intuition in many ways. Like we think, between these tiny bacteria and us with all of our capabilities, there must be a huge difference. Not so huge, just 4 or 5-fold. Any questions about that? Comments? Anything you find surprising? There is something for me at least surprising, because it means that we had way more space in terms of what of place to code stuff on the genes. Evolution was it's there, and still we're not using it so much in some sense. So there's something to be explained here. There's about a factor of about 100 between what you between the genome length and what is being coded within it in terms of genes. And the question is, where does that go?
Junk DNA.
Okay, so let's throw out all sorts of suggestions. So I hear junk DNA.
Introns.
Junk DNA. Then I heard introns.
Transposable element.
Just somebody else saying something? Transposons.
This may be like the same as transposons, but like viruses, ancient viruses that have infected us.
Regulatory.
Yeah, regulatory.
Regulatory regions. Anything else?
Pseudogenes?
What?
Pseudogenes.
Pseudogenes. Good. So I'm taking this opportunity.
A more fixed genome.
What?
A more fixed genome. You're not having like thousands of gene transfer to chuck stuff out.
Good. So I'm taking this opportunity both for us to learn some biology. So we could talk about what is each of those things. But I think it's also like it sharpens the mind in a way for me, because like, you know, when you have this quantitative view and you see this discrepancy, not only you could learn about new stuff in biology, but then you could look at like, okay, you know, how dominant are they? What's really a big contributor here or a small contributor? So briefly, let's talk about that. You know, you can obviously dig much more deeply into that. There's a thing called transposons, which are, you know, jumping across they could jump across the DNA. They kind of like, or hijacking parts of the genome and could replicate, etc. And that's about half of the genome in humans. So it's quite amazing that all of those transposable elements, so there's a sign and line and all of those things, different variants of things, they could be about half of the genome, which is a lot. It's half. At the same time, it's not a factor of 100. So that explains part of it. And viruses are related, and pseudogenes are like, kind of like, except for those 20,000 genes that we have all sorts of other genes that, you know, duplicated at some point, and then, you know, something happened there and they're not functional anymore. Like the start codon doesn't work anymore, sorts of things like that. So there's also a fraction of those, but that would not give you, that's not half of the genome. So in general, you see the coding genes are about 1%, right, from the 100-fold that you see here. So that might add like another percent or something like that. What's the other big contributor out of those things? Okay, so let's talk about introns. So we talked about this as a very big contributor, and that's related also to other stuff. Introns, what anything knows about, you know, what would their fraction be in the genome?
For every gene, it's more than half sometimes, right?
Right, so for every gene that you have, it's broken down into different parts, the exons, and the introns are on the average about an order of magnitude longer. So if the genes take 1% after you look at the order, with the introns it would be, say, 10%. So it's about 10-fold more. And that's really a much larger fraction than what is actually being expressed in the end.
Aren't the introns considered as part of the genes?
Right, so that's a good point. You can think about them as being part of the genes. So in that respect, really, what we see here is a factor of 100. That's like if you were just looking at the expressed part, but then the difference, because the gene length, instead of being 1 kilobase in bacteria that we said are pretty compact, when you include also the introns as part of the genes, you'll find them to be much longer, about 10 or 20 kb. Good point. Okay, there's more to be discussed, and then junk DNA might be related also to regulatory intron. Junk DNA is like a term that we gave to the fact that like we saw that it's much, much longer, and we don't really know why we have this 100-fold. When you start to understand that there's these transposons and introns, etc., the term junk doesn't seem to fit so much. And it's an interesting question whether in the end you should call it junk or not, and I would say it's still kind of like an open question out there. What fraction in the end would really be junk? Where junk is just the fact that, you know, evolution might not be paying a very strong price for increasing the genome length. Like if you're already 3 billion in length, maybe it's not such a big deal. If you'll be 3.1 billion, you should look at like the cost benefit. Like, you know, are you gaining anything from it and what are you paying? It seems that bacteria, evolution is telling us that bacteria are paying a strong price by the fact that it keeps everything compact. In eukaryotic cells, especially say mammals, but also plants, etc., it seems like there's no strong selective pressure, and therefore the genomes are pretty large. And actually, we're not holding any record. There's all sorts of, you know, rice has a longer genome, as far as I remember, from us. And then there's other things like some amoeba that has like 100-fold larger genome still, like hundreds of giga of base pairs, and also some very small plants. Okay, which is also kind of like not completely intuitive. Good. So now let's move into talking about these genomes in terms of, you know, how evolution plays around with them. And for that,
Can I ask a question?
yeah, sure.
So like in prokaryotes, like in E. coli, it's really like replication speed is really important when you're talking about evolution. Has any work been done to like characterize, like can you probably make the calculation for what the impact on replication on like eukaryotic like cells is, to see like what the impact on replication speed in the eukaryote is for with all this not junk, but like
Fantastic.
extra.
So if I got it right, you're saying the following. We know that in bacteria, which replicate relatively quickly, and then it's super important in terms of selection pressure, like the competition is about the fitness, who is the first to replicate. The genome length is what they need to replicate, and that could be a limiting factor. And the question here in like eukaryotes or mammalian cells, etc., where you have 1,000 times more longer genome, let's say if you did it the same way, you might have thought that it would be 1,000 times longer in order to replicate. And that would be really bad, because if a bacteria replicates in an hour, these cells would take 1,000 hours, which would be over a month, and that would be really, really slow. So what's the solution?
Compact genome.
No, but they're not compact genome. I'm talking now about those long genomes, just like us.
Multiple
Faster
application
RNA
ports.
polymerase.
So maybe faster replication. So if you remember, the DNA polymerase is already pretty fast. It's 1,000 bases per second. It would be very tough to make it way, way faster.
It's multiple replication.
So we need a different solution. But it's good. I like the way you're thinking, like you're connecting here again. When you put the numbers, you can understand, okay, there's a puzzle here. There's some problem that nature has to solve.
So,
Or maybe we're stuck. There's like no
We're
solution.
just dividing ourselves less.
Small polymerases.
Okay, more polymerases.
division.
Okay.
Everyone will have shorter piece of.
I love it. Okay, so that's the solution, exactly like you're saying. So really nature decided also to do like divide and conquer. Instead of saying a polymerase, okay, you have to do the whole genome, kind of like the way it's being done in bacteria. Now let's just say, each polymerase, you don't have to do everything. You just copy, replicate your part, and that's enough. And then you can work in parallel. It's like parallel computing. You can do 1,000 different polymerases working in parallel, and then you're fine. There's one challenge though. Yeah, you need to piece all of that together. And nature found a way to do it.
Chromosomes.
What?
In humans, it's have few chromosomes.
Yeah, so that's another thing. You have several chromosomes, so it's broken into chromosomes, but still each chromosome would be like 100 times longer. So even within each chromosome, the several, as you were suggesting, several polymerases are working in parallel, and now you just need to stitch them together. And I think somebody got the Nobel Prize for understanding exactly how it stitches together, but I don't know the molecular biology of it right now. But you understand that like it's dictated by the constraint of the rates and values that's associated. Good, so thanks for that question.
Also, our cells divide less, right?
Yeah, so they still divide less, but not to a factor of like how much longer they are. Okay, it's also that's a good indication like of how, if we just keep the discussion at the level of qualitative statements, you would ask the question, and then this could be the answer. Yeah, but they're also but they're replicating slower, and then you say, so no problem. Or maybe somebody else say, yeah, but there's chromosomes. So the chromosomes are dividing more slowly, and everything is fine. But actually, if you put the number, it's like a 100-fold or maybe more, and it doesn't work out. Or 500-fold, right? So 500-fold, you can't do 500 just with the chromosomes and somewhat slower. Okay, but things are going to get even more surprising, I hope. So let's ask the following. After a bacterial cell just a second, let me think how to ask it correctly. How many mutations are accumulated in the progeny of an overnight culture. Okay, so I'm thinking here about what maybe some of you did in the lab many times. you put cells for an overnight, like you take, one cell or a few cells, you're putting them there, you let them divide throughout the night so as to have many cells, and then if I would go and sequence the cell at the end, in the morning, after the whole population expanded, and I'm comparing between that and the cells I started with, the question is, if I look across all of the genomes, how many mutations accumulated?
Are we talking about E. coli or humans?
Yeah, let's talk about E. coli. You can try and do it with humans. It's a question what kind of lab we'll have to do it. So let's talk with E. coli, and let's say it's A, it would be about 100, B, about 10, C, few, and D, much smaller than 1.
But that's for the specific bacterial progeny or the
Yeah,
whole population?
so let's start with, I'm looking at the progeny that I'm sequencing one in the morning, I'm comparing it to its ancestor.
One cell.
One cell. Anyone votes for A? Few people. B? Half of the people. Few, 2 times few, less than 1, 1. Okay. Good. So let's put this question just for a second aside and ask the following. In one replication of E. coli, how many mutations are created? Question is clear? Now I don't look at the overnight and all that. I'm just saying, I'm taking a cell, I let it replicate its genome. I'm saying how many mutations are in the new cell, in the daughter cell. Okay? Quick vote. About 100. About 10. Few. Less than 1. Good. So how do we go about, answering this? Anybody remember something about mutation rates by any chance?
It's like 1,000 base pairs per replication.
Okay, so I think in the key numbers that you had, it's like, I think we had something about mutation rate. It's actually a pretty useful number to carry in your head. So in E. coli, it's quite similar in humans as well, which is also interesting. It's about 10 to the minus 10 mutations per base pair per replication. That's a very low number. Okay, 10 to the minus 10. And then it's mutations per base and per replication. Okay, which means that if I want to answer this question, and this is, by the way, thanks to the fact that not only, it does what it does, it also has this thing called proofreading that ensures that, if it did a mistake, it actually looks again, checks itself, and makes sure that it's actually pretty astounding. Like if you think about 10 to the minus 10 errors, it means that you can write the Encyclopedia Britannica with all its, and you make 0 errors. You can, you could do like that. It's like a very, very low error rate. Like your error rate in anything you do is way, way higher. Okay? Anyone that ever would achieve anything close to this, I'll be, Okay. So let's go back to this question. How many mutations are occurring? So if this is the mutation rate, what do I need to do next in order to understand how many mutations I'll have? Yeah, so I need to get rid with the bases, right? So how many bases do I need to multiply it by?
5 mega.
Yeah, so 5 mega. So let's even make it 10 mega, because actually I think this is per base, not per, I'm not sure if it's per base or per base pair. And you have, maybe you're doing 2 of them because you're separating the DNA. You have 5 million here and 5 million there. So let's just make it 10 to the 7, like 10 million. So we're getting about 10 to the minus 3 mutations per replication. So it means that if I sequence it, no mutations. It would, vast chances that the one I'll be will have be exactly the same. Okay, that's great. So we finished with this thing. Now let's go back to this one. So what did we have here? I took a cell, I let it replicate overnight, and then I was looking how many mutations I had in the morning. Okay? So this is nice because now this was mutations per replication. Now I need to multiply by the number of replications. So how many replications?
30?
Around 30.
Okay, so we had the gut feeling here of 30. Anybody has a different gut feeling?
It's not a gut feeling, I know it's 20 minutes for application.
12 hours.
I give it 10 hours.
Okay, good. So what you're saying is it's not just a gut feeling. It's actually evidence-based gut feeling. So let's see, there's different ways to do it. I didn't think about that way. I like it. It's a very practical way. Maybe it's because I don't put overnight for, forever. So, okay, so the way is being suggested, we're putting it for overnight. We need to know the cells are dividing. How fast are they dividing? Who knows? About, say, every half an hour. Or let's say every 20 minutes. Let's say every 20 minutes. So that's 3 replications per hour. And if you were waiting, if you left the lab late and you come early, so just 10 hours, so that's 30 replications. That's the logic, right? Okay, that's nice because actually I was trying to push you to say maybe somebody else has a different intuition. Instead of 30, maybe it's like 3,000 or whatever. But it can't be. Okay, in 10 hours you can't do 3,000 replications. Good, so you could take this and multiply it by 30 in order to answer this question. So we'll have, we could just do it here, times 30, and it will still be much smaller than 1. Which is interesting. It's not only so now even if I take, I take one of the cells in the overnight, and then I look over all of its genome, it would look perfect, just like the original one. Much less than 1.
If you ask how many mutations accumulate, don't you mean the entire population?
Okay, so the way I asked it here was about like I look at a specific cell and I'm sequencing it and I'm comparing and this is the answer. I think you're asking an excellent question. Let's get back to it a bit later. Good, so if we did all that, I think we're ready to try and apply it further.
Did you just calculate the probability of seeing a mutation basically in after overnight growth? Like
Can
in
you
one
repeat
cell?
that again?
You basically calculated the probability of seeing a mutation, or if you grew like a single mutation in a single cell.
So I wasn't looking, so I didn't look at a specific mutation. I was saying any mutations anywhere on the genome, and we saw
in a cell.
In a cell, yeah. And we saw that like I was choosing a cell, it would be just like its mother or grandmother 30 times.
So to know like the number of total mutations in the population, you would just multiply by the number of cells?
Yeah, so we could talk about it a bit later, yeah. Okay, so let's go back, in time and talk about SARS-CoV-2. You remember once, some time ago, we had some pandemic in the world. Anyone remembers there was also an issue there with mutations? We started having variants, right? When looking at so we're talking about SARS-CoV-2. Anybody remembers, what was the first major variant hitting us?
Delta.
Beta.
So was it Delta or Alpha?
Alpha.
Yeah, so Alpha sounds like it should come before Delta, yeah. Okay, so we did have the Alpha variant. And then there was an interesting thing with Alpha. It had lots of mutations, right? Being a variant, it was quite different than the original strain from Wuhan. Anybody remember something about how many mutations there were there?
2 words, a few.
Yeah, so people, usually expect, say, 2 mutations or a few. But then it turned out that when looking at the Alpha variant, it wasn't 2 mutations. It was about 30 mutations. And then the question that I want to pose is, does it make sense? And what does that tell us? about, the origin of this. Does it require some special thing in order to achieve such a mutation rate?
When you say 30 mutation, is like 30 single nucleotide replacements that changes
Exactly.
the entire.
Yeah, that if you take the whole genome, anybody knows the length of the genome?
5,000?
About 30,000. So if you look at the 30,000 and sequence it, there's about 30 different mutations along the genome in comparison to the strains that we had before alpha. Or at least the original strain, like in the sequencing in the beginning. So there's two ways to try and approach this, looking at this. One is more microscopic and one is more macroscopic, okay? Microscopic, you can do kind of like the thing of like what would be entailed in looking at the number of mutations that you expect to see, say between the ancestor that appeared first in China and what you saw here of alpha. You need to know the mutation rate. You need to multiply it by the genome length. And you need to multiply by what? By the number of replications. And that's fine and well to do it this way, but I would say it's not so easy because the mutation rate, these are viruses. Viruses could be pretty hectic in the mutation rate. It's not like the number that I gave you for bacteria that is similar in humans, that is relatively constant. With viruses, it's kind of like the machinery doesn't have proofreading. I think it doesn't have proofreading. It's more complicated. What this does? Okay, it's good I corrected B before you corrected B. But yeah, okay, so I don't remember exactly how it works, but there's this. Genome length we kind of know, although maybe many of us don't know. So I actually want to give you tools that would, instead of you needing to remember many numbers, I want to have few numbers and something inferring from that. And then the number of replications, that's actually the most tricky part, or also a tricky part to try and infer that exactly. Okay, but this is I could do it that way. Actually, Ron Sender was here, like we did it during the pandemic, and we shared it with the world, and that gives us some insight. If anybody's interested, there's a cool thing about like the key numbers related to SARS-CoV-2. It's been translated to like 30 languages. It's all fine and well. But you can do it also in another way. And that's kind of like easier. You could say, okay, let's take and just use sequencing of between the virus that you take from somebody, and if that somebody infected somebody else, you can also take the somebody else and just look, okay, how many mutations are here for one generation of infection. Okay? So you can also look at the mutation rate in units of per viral genome. It's not that way. Per viral genome and per generation it's called. How do we? Yeah. You can see here I'm saying generation and not replication because it's different. You have per infection. Infection generation. Okay, because what you have is that like the virus replicates many times inside the cell, and then after that happens, many times, it's like an overnight. We're serving like an overnight for the virus, and that jumps into a new infection, and so that infection is like the generation time of the infection or overall process. Okay. Okay, and that's actually easy to measure. It encapsulates within all of these numbers, and it comes out to about 0.5 mutations per infection. Okay, that's overall. So that's if I looked overall over the whole genome, within a cycle of infection. What?
between persons.
Yeah, so by that I mean between persons. So is that not exactly infection? Infection is within.
Just to be sure that it's not
Yeah,
like
so
between
yeah.
cells in the same persons that we infected, like between I got infected and now I'm passing out to you.
Thank you.
So now you will have additional half a mutation.
Good. So yeah, so if I was I got the virus from Ron, I would have half a mutation more than he had, because in the whole process of what happened for the virus in his body in this big overnight, we got about half a mutation extra. Okay, if you remember it with the E. coli and overnight, just to do the comparison, you remember it was 1 in 100 for the whole genome on one replication. Then we said there was about 30 times, so it's 0.03. So it's about 10-fold less than between our infections. Yeah.
So, to make things short, I sequence at the moment that the first person was infected in the first place, and then I sequence the other, not between near the time of the infection.
Yeah,
right?
so,
Okay.
yeah, so that's a good way to think about it, near the time of the infection, but actually it wouldn't be very different if you just take some random
before the
time.
overnight or after the overnight.
Yeah, but if you take roughly at the same time, so let's say even that you just, you did a swab from Ron infected me, you take a swab for me, let's say it's roughly at the same time, there's half a mutation. And it actually doesn't matter so much, unless you took it just when it was exiting Ron and coming to me and immediately, but you can't do that, because you can't do all sequence when it tells I have one, I'm not sick yet, etc. Okay, so far so good. Okay, so we have about half a mutation per infection. So how do I get between that and the alpha strain? Okay, so there's a question about, how many infections cycles there are. So there's this thing called the generation time, which is the characteristic time between Ron getting infected and Ron getting infected. Okay? Anybody knows anything about, anybody remembers about, what happens with that in COVID-19?
Like 3 to 5 incubation.
Right, so it's usually a few days, it's on the order of like 5 days. It also changed with the variant somewhat, etc. But it's about 5 days. Good, so if I have about 5 days of generation time and I have half a mutation per infection, so every generation time is one infection, I can combine these two. So what's an easy number to remember? Let's think per month. Okay, per month, 30 days. So if you have 5 days, how many generations would I have in a month? 6. So 0.5 mutations per infection times 6 infections per month. So we talk about 3 mutations per month. Good. Anybody remembers how long did it take the alpha variant to arise? Or let's say, did anybody have here, remembers getting infected or somebody else getting infected with alpha or reading about it? When did that happen?
January 2021.
Yeah, so that was somewhere around December 2020 or January 2021. Okay, and if we're comparing it, when did the first sequences of the original strain came about?
No.
So
December.
yeah, so it's just the same a year earlier. So this was like December 2019. So we're talking about a year. How many mutations would you expect to accumulate within a year?
30.
Yeah, so there's about 10 months in a year. At least in this course. So actually 30 mutations is what you would expect just by random, what's known as normal drift. Okay, even if there's no selection pressure, even though clearly, if the virus, becomes more infective, there's a selective pressure for that. But just in the normal rate that we find of mutations, that would give you 30 mutations a year later. So there's nothing crazy special happening. It doesn't have to happen, I don't know, in a lab. It doesn't have to happen in an immunosuppressed person, which is also one of the mechanisms that was people were thinking about. It's just the normal rate of mutations. Maybe it did happen in the immunosuppressed person. It's not that I know that, but it's not necessary.
So isn't this number surprising because we would expect it to be a shorter time because of the selective pressure?
Okay, so the question is, isn't it surprising, like maybe because of the selective pressure, you would have expected a faster mutation rate? That's what you're asking?
Yeah, but I don't see how it can accelerate the mutation rate, but
yeah.
the
Okay.
lack of a more effective...
Okay, I think you have a good point here. It actually would also connect to what we'll be talking in the next hour. So I suggest we maybe get back to it. I think it's a good question and not easy to answer. I'm not sure I have a perfect answer about that.
I think it's mainly that like this is your regular mutation rate, but it's the selective pressure that lets this specific variant rise and actually become dominant. Because you'd have many of these, almost every combination possible of the 3 mutations, but there's only one that's actually rising because of the selective pressure.
Yeah, so that's also true. That definitely this strain we know, it's no surprise. It's not a coincidence that became the most prevalent strain in the world until a better strain arrived. It did have a fitness advantage, and that's why out of those many mutations that accumulate, this was the strain that actually got selected, which doesn't take away from your original question, which I think is also valid. Okay, I think with that should we pose let me just pose the question that would come after the recess, and we'll go from there. By the way, I think there's an interesting coincidence here that I was just thinking. So remember we calculated that for the E. coli bacteria that we're doing overnight, the number of mutations accumulated was about, we said 0.03 in comparison to 0.5. It's about just over tenfold difference. But then if you take the generation time of 5 days versus 10 hours for the overnight, it's also about the ratio of 10. Which in a funny way, it means that like the rate of accumulation of mutations in bacteria and in these viruses is actually pretty similar, it seems to me. Unless I missed something. Which is kind of a coincidence, maybe, because it's like different. So it's all related to its different genome length and different, rate of replication. But in the end, it comes out to be quite similar. I need to think more about it. It's like this 10 to the minus 10 and 10 to the 7 and all, and the rate of replication, everything somehow fits together to give you such that also in E. coli, if you do overnight after overnight, let's say you finish the overnight, you put it for another overnight. You put another overnight, you'll also get something about roughly the same rate of mutations. So about on the order like 1 per month is kind of like seem to be similar across these domains of life.
But the density of mutation is much lower.
Right, but the density of mutations is much lower. That's a good point, because this was like for the whole organism, for the whole genome, which was millions of bases, and now it's just a few tens of thousands of bases. Okay. So, with that in mind, let's ask the following. I want to add in the issue of selection, okay, or function of mutations, okay? So now here's the thing. In my lab we work a lot with E. coli, and sometimes you want to have a very specific mutation. Okay, maybe this is the mutation that would give you the ability to utilize carbon dioxide, or to change your metabolism in a different way, or maybe create some special compound or whatever. And now the question is, if I want to get a specific mutation, let's say it's like on the genome, it's like in base 2, 3, 4, 6, 9, 7, 2, and I want like a guanine turning it, something into something. And let's say I also have, I can select for that. Like let's say I'll put an overnight, and then with the bacteria that I'll get, I can see if I have now the mutation or not. So in order to achieve that, I could suggest 3 options. One is to use something called site-directed mutagenesis. Which is kind of like how, it's kind of a poor man's version of CRISPR. Like I would somehow target this specific location and I would tell the machine, okay, change this to that. Okay? Then there's another option, which is, I forgot the name. Yeah, I can do some random mutagenesis. Which that means, I'll just throw into my, so this, I'm going here like an overnight. So that's like, 5 ml tube with bacteria that I let it grow. So I can throw in some other mutagens, some chemical that would increase the mutation rate by a lot. It could be by like factor of 1,000, 10,000, something like that. So that's my second option.
Is it independent of replication, the mutagenesis?
What do you mean?
So the mutation, like if you do UV light, it would be independent of replication, I guess?
I think so, but I don't know enough about it, but it's good that you help me. So an example is just put it on the UV light, like let them sit in the sun, kind of like without putting anything against that. If you put the bacteria there, they're accumulating way more mutations. And the question is whether the mutation, I think it is dependent on replication. If there's no replication, but that depends on the mechanism, different rates, whatever. And the third option is, do nothing. And the question is, what do you need in order to get that very specific mutation? Question is clear? Somewhat?
So it's not only this mutation, but at least this mutation, right?
Yeah, at least this mutation. Okay, so I suggest, we let you think about it. We'll go for the break, but it's going to be a special break. It would be like a 10 minutes break, but within that break, I also want us to meet in the lobby here and to have like a group photo of the course. It's something we're doing every year, and it's something that's being left as a nice leftover that you took the course. So we have 10 minutes, let's meet there, we'll take a photo and then drink or do whatever you want. Okay, everyone, let's get back to where we were. We were looking for this very specific mutation in E. coli, and the question is how hard do we need to work in order to get it? Good. So we already know something about the rate at which mutations are being created in E. coli, right? What was it? It's about 10 to the minus 10 mutations per base per replication. And now I'm looking for a specific mutation. So the fact that this is per base is what is relevant, right? So I'm not multiplying by the length of the genome. But I am going to multiply by the number of replications. But here there's a really delicate point here. Okay, so bear with me for a second. Before, you remember, we started with some cell. And then we put it for an overnight. And we had many generations. And then we looked at the progeny and we compared it to the ancestor. And we looked at that and we saw that basically this genome would have 0 mutations in comparison to this if you had to choose one number. But it was actually 0.03 or something like that. Okay. Now I want to see if within my 5 ml, I'm looking for a specific mutation, but it doesn't have to be in this one. It's also okay if it would be in this one or in any of the others. So now I'm actually thinking about how many replications I had altogether. Okay? Or in general in the progeny. The difference is clear? Okay, conceptually it's not, it's easy to think about the difference. Quantitatively, it's a huge jump. Because here I'm looking at one progeny, and here I'll be looking at lots of progeny. How many progenies?
10, hundreds of millions?
2 to the power 30.
2 to the power 30.
Okay, so one way to think about it, I like it, is 2 to the power 30. If we said we had 30 replications, to 2 to the power 30.
You allow yourself the specific in the way, like not in the final generation.
Yeah, I'm looking for, because what am I going to do? I'm going to take my 5 ml, and I'm looking for a specific mutation, let's say gives me resistance to something, or create, so I'll just I have a selection assay. So I'll just throw them on my plate, and I would see if I have a colony, meaning, and that's enough that there'll be one bacteria that would have that very specific mutation. How much is 10 to the 30?
Giga. 1 billion.
What?
1 billion.
1 billion. How do we know this? Because there's a nice way to do it. It's 2 to the 10 to the 3. 2 to the 10, that's about 1,000. To the 3, so that's 10 to the 9. Okay? So that was done, in the most crude way just because you said 30. Okay? You can do it at different levels also of so let's do it a bit more deeply. What's gonna sit inside here? These 5 mLs.
Optical density.
Yeah, so this thing with optical density. So how much, let's say I have an optical density of something, of a few. How do I transform that into number of cells?
It's a billion per milliliter.
Good. So it's good to, if you're not in that field, it's not such an important number. But if you are playing around with growing cells, bacteria, etc., it's good to carry on. But if you're growing other cells, there's about 10 to the 9 cells per ml per OD. Okay, so that means that if you have 1 ml at an OD of 1, you have about 1 billion cells there of bacteria. Okay, now here what do I have? I have 5 ml. OD after an overnight, let's say you reach an OD of 2. So 2 times 5, it's 10. So I actually have 10 OD units here. So it's not 10 to the 9. I would say if you do it more accurately, you probably have here something like 10 to the 10. In short, it's kind of like the same. It just shows us, this 30 was actually a good ballpark. But actually, there's also this thing that with exponential growth, it makes a difference if it's 30 or 33. 33 would already bring you here. Okay, so you're kind of sensitive to this number. But still, we see it's also nice, the 30 was very helpful for us in the previous analysis, that we knew that if you started with 1 in 1,000, 30 generations, you still have less than 1. And it doesn't matter if it's 30 or 50 or whatever. If you're talking about, if you really care about the number of progeny, then it becomes a big difference if it's 30 or 60. Okay, so it shows again, when do you need to care about your accuracy? Okay, so taking all of this and going back to this mutation rate, what does that tell us? It tells us that, let's go with the 10 to the 10 for a second. It means that actually in terms of number of replications that I'm going to have, I've had on the order of this number of replications. Right? There's different ways to do it. You can say, how do I do exactly? Yeah, it's actually, if you started with 1 cell and you're finishing with 10 to the 10 cells, you had about 10 to the 10 replications.
The rate of death is not...
What?
The rate of death of cells
The rate of death.
is not very relevant.
I don't know. First of all, they live in this heaven for bacteria. They're in LB growth. They're happily going. There's some fraction of them that might be dying, and that could also have some effects. Okay, so this brings us to about 1 mutation per base. So in principle, it's very reasonable that any mutation you're interested in, you'll have it there. And you basically don't need to do anything special. Maybe you need, instead of growing 5 ml, maybe grow 50 ml just to be on the safe side. Which shows really the power of so every time you do this thing of growing a culture of cells, just in a small tube, you're actually covering all the genome in terms of all the possible mutations.
Not really. Find a specific cell with a specific mutation.
What?
You need to find a specific cell with a specific.
Exactly. So it's interesting. You have all the range within there, but now if you're looking for the specific one, you need to have a good selective, like a good way to detect that specific cell. If you don't know how to detect it, you're in trouble. If you have a good way to detect it, you'll get it.
So if your mutation happened early in the replication cycle that you're running, then the chances are your bacteria probably maybe died or something happened.
Okay, so there's an interesting point here. I was so like, you could think what happened if your mutation didn't happen here. Let's say it happened here. So now it's all, the whole tree that came out of this one would have that mutation. Now it wouldn't be one bacteria, you already have like 10 to the, I don't know, 10 million, 100 that will carry that. That's also true. But I think it doesn't change the overall thing. You can think about like either
half of the replication are at the end.
Yeah, half of the replications happen just in the last generation, and then another quarter in the previous generation. There's all sorts of fun stuff you could do with it, and you can infer all sorts of properties, but it's beyond what we have for today. Okay. That's, by the way, like I find that like many people who are deep into like this is something that is kind of like both useful and surprising to many people to understand that like actually you reach such a large variety inside when you're growing cultures. It's all a question of numbers. Okay, so now let's try and use that to go beyond bacteria into humans. So let's say I want to have a specific mutation in order to save humanity from, I don't know, for Ebola, you need to have a specific mutation somewhere on the genome in order to give you resistance or something along those lines. So now it could be, it doesn't have to be this because now we have a much longer genome. It could be this, what did I do here? It could be this specific mutation that I'm looking for. And now the question is, let's say I want to have this mutation in the human population, de novo. The question is, how long do I have to wait, or where would I find it? So how difficult is it to find a specific mutation within the human population? We could do kind of like a similar analysis, right? Because we just said the rate of mutations is also 10 to the minus 10. Mutations per base per replication. So now if I want a specific one, I could just look at the rate of mutations, and now I need to multiply it by what? By the number of people that we have on Earth, which is about how much?
8 billion.
10 to the 10 people. So you see, I actually have a lot of representation of mutations, every specific mutation somewhere in a population. Is that enough, or is there something that we could add on top of this?
You know that you have areas that are generally way more conserved than other areas in very big orders of magnitude difference of how conserved it is. I don't know, it seems like it's fair to ignore it, but it's still weird.
Okay, so how does this plays into this? The fact that like there's conserved areas. Anybody has a suggestion?
Some mutations you'll find a lot, some you won't find.
Yeah. Also, if you look in areas in which there are Ebola, maybe you find more of the relevant mutations because you've selected.
Okay, so I think in most cases, this issue of like 10 to the minus 10, mutations per base per replication, that actually happens irrespective of whether it's a conserved area or not. Because this is like a molecular mechanism that like it's very tough to do much better than that. But
people with those mutations.
Right, but then what you'll find is that these areas are such that it ruined some very important gene, And then people that got that mutation, they wouldn't survive, or there wouldn't be good development of the fetus or something like that. And therefore, that's what makes it conserved, is the fact that this is not leading to a viable progeny. Okay, so that's how you're getting the conserved areas. Yeah?
Two things. One, are you looking for like a living person with that mutation? Because if you are and you're planning on, I don't know, PCRing everyone, then you should definitely go for it somewhere with more diversity. And two, are you considering that, like, if you're scanning every mutation in the human genome, that like some number of them, and I think it's not a very small number, are, like you said, they're not leading to a living human, and therefore should be excluded, maybe? Because some
Okay, yes, so that relates also to the previous question about conservation, etc. And that's true. There is a process such that, only a fraction, but I think that's actually, it's a significant fraction that, that passes through. But there's something else here, just, yeah?
People and replication is not the same unit.
Exactly, okay, so good point that you noticed. We're talking here about per replication, but a person coming from a different person, just before I had my father visiting us here, it's not like, it wasn't one replication of the genome to lead from him to me. Okay, there was something else happening along the way. So the question is, how many replications from your parents to yourself?
It depends which parent.
Okay, first of all, it depends. We like this answer a lot. But you're saying more than that, you're saying it depends which parent.
And because the egg is arrested for.
Okay, so let's talk about mothers and father. So how many mutations or how many replications from your mother's side and from your father's side?
Can you explain the question?
Okay, so I'm saying we calculated here mutations, but this was just like, we talked about the number of people, but this was before talking about the fact, okay, how many, that was per replication. But actually I have quite a few replications per generation, right? And we really care about, the rise of mutations after a generation. Or at least I'm interested now. And so the question is, how many mutations, how many replications arise in transitioning from a parent to the child?
But where do you start counting and where do you end counting? Because
okay, so I want to look per generation. So let's say from when let's say in the kind of like what we were discussing before, let's take it at the same time. Okay, let's say at the same age, like when my father was just born and when I was just born, or when my father was, my age now, or anything along those lines. So you get the rough value per generation.
It would be easiest maybe to think about the zygote, and from zygote to zygote, I think.
Okay, so really I agree with you. It's not so much about, person to a person, because like what really gets transmitted is the zygote. So, either the sperm or the egg. So let's say between a sperm and the sperm, an egg and an egg, right? The zygote to the zygote. Okay? Actually, it could be, yeah. Okay, so should we do, so how do we handle that? You could look into the details, but any gut feeling of how many would it be?
50.
I have 50. Yeah, so I think it's about 30 replications, which you can really derive from looking at how many eggs are being formed and things, and a bit more information about that. What about fathers?
Much more. Much more.
So it really depends on the age. If somebody gave their sperm led to birth at an early age, it's actually also would be pretty similar, like 30 replications. But if it was at a much older age, it would be about 500 replications. That's just because our sperm is being formed continuously in our life, and so it's very age dependent. So if there's something you really like and you have a new mutation in yourself versus your parents, you can go back to your father and say thank you, because probably it came from your father. If it's something you don't like, it's also because of your father. Okay, so this is being led to a large extent by that, especially with age. Okay, that tells us back again that in terms of like, if you're looking for some specific mutation in the human population, okay, I didn't explain it too clearly, but what I meant is like I'm looking for a new mutation that would appear in the current generation that didn't exist in the previous generation of humanity. Then we shouldn't, we know that somewhere among the people of the earth, because that's the rate of mutation, that's the number of people, and that's the number of replications, I'm going to have several dozens of people that will be carrying every mutation, unless it's somewhere specially conserved, and therefore it would be selected against in the process of, giving birth or development. Questions, comments? Okay, so let's go from here into the next stage, which is, it turns out, it's nice that we got this, but now we actually need another mutation. And we need T to C. Now we need 2 mutations. And now I didn't have it in the previous generation, and now I want to have it in the current generation. What's the chances for me of finding representatives of that?
2 to the power of 2.
What?
2 to the power of 2.
So now it's kind of like something to the power of 2. I could ask whether, I would again have it available, let's say in the population, several, of the earth, or I need to wait a while. So you can think about it that way. In the one generation that I had, we did a calculation, let's say I had, 30 people or something that had the first mutation. Now, instead of having 1 billion people, I just have 30 people, and I need to have the other mutation. The chances for that, we need to do the multiplication again. It's 10 to the minus 10, but now instead of multiplying by 10 to the 10, I'm just multiplying by a few dozen people.
But can't you have both mutations in the 10 to the 10 generation?
I can. That's what I'm looking for. I'm waiting to see if I could get both mutations. But they're happening independently. So I could just say, okay, what are the chances that I'll have both of them? I could just say, okay, I'll need to get the first one and also the second one. Just the first one's going to leave me with just a few tens of people. And then the second one, the chances if I'm multiplying 10 to the minus 10 times 30 or 100 or whatever, it's tiny. I would actually need to wait. So if I need both mutations, I would need to wait millions of generations, millions of years, until randomly I would get 2 mutations. So basically, this is never going to happen, like in real life, for humanity. It's way more than the time humanity has been around. Which is kind of, I think, interesting and non-intuitive, that on the one hand, if you want to get a mutation, either be it in E. coli or in human population, no problem. You have everything you want available. But if you need to have 2 mutations independently, you're not going to get it. Maybe in bacteria, if you have a big enough, maybe at some point you'll get it. But then if you need 3 mutations, forget it. And I think it's especially interesting in the sense that in spite of that, we developed from something like bacteria with many, many mutations that were selected for. So what does that mean?
Selection.
Random, but you take the equation from 2 variables, you combine. You don't have to wait for all these millions of generations to get the 2 mutations together. You just find the 2 people and their kid will have both.
Okay, and you were saying?
Cancer is evolving with more than 2 mutations.
Cancer is evolving with more than 2 mutations. Okay, so there's several things that have been said here. Okay, one, somebody said something about random and non-random, like there was a discussion about random and non-random. There's been a discussion here about sex, about like, the fact that you're combining things from your mother and father. And there was a mention here about cancer and the fact that in cancer you have several mutations happening in the same person leading to cancer. So, first of all,
relatively rare that you need a very specific position to be
what?
mutated? Isn't it relatively rare that you need a very specific position to be mutated?
Okay, and then there's a,
Have
yes.
different options.
Okay, and then here there's a comment about the requirement for having a very specific location. Okay, let's write it for a second. So the comments that we had here was about random, about combining sexes, or what is the right name for that?
Recombination.
Yeah. Recombination is something, a very specific mechanism, but there's also combining. Then there was issue of cancer. Then there was a question whether we need specific. Maybe there were other comments. Okay, what I want to say is that it's actually it's a complicated question in the sense that there's several things happening here about how evolution really works, and what we were doing is just like showing like the first, like the key power or like of how it works, but then things come into play. Okay. One is in relation to being random. So what happens is the mutations are random, but then you have selective pressure that chooses them. So if I need to get two mutations, as you saw, next to impossible. But if the first mutation would already be giving you a selective advantage, that maybe, let's say, you have these 30 people, staying alive, but then now if this was, you let them expand. Now when you do again the thing that you need the mutation, you have again, let's say 10 billion, you could choose that, and then again you have everything you need. So if every mutation by itself gives you a selective advantage, you can do as many, like you get one mutation, then you get another. Every time you need the mutation, you have it. If you need the combination, that's the tough thing, if they're not giving you each one a selective advantage. So basically you need to work such that everything would give you, every step would be useful. Or that something would also be hitchhiking, or there's all sorts of other mechanisms that could come into play. There is this that's also useful, like if your mother got one mutation that is beneficial and your father got one mutation that is beneficial, and each one is now propagating in the population, now sex is a great way to combine beneficial mutations. So that's also very helpful. Cancer. So in cancer, usually you need something like 5 mutations, they say, or something like that to give you. So how does that work?
What you said before, the fitness advantage.
Yeah, so you need each one to have some fitness advantage so that it would expand. So it's true that for the full repertoire of what it takes to have a cancerous cell line or transformation, you need several mutations, but usually each one would have some advantage along the way. It also relates, so the other point was, it was like the specific mutation. So it's true that also sometimes it doesn't have to be a specific one. It could be, you'll have like a target. Say, I know, in cancer, you might have a mutation in P53 or in MDM2, some transcription factor, or it could be some receptor or whatever, that, many mutations that would happen within this target, which would be, let's say, 1,000 base pairs or maybe more. Many of those things could lead to something that would, ruin the enzyme activity or the protein activity, and therefore would give you an advantage. And then you have many more targets that you can hit, and by that you don't need the specific mutation. And all of these answers are, correct and part of understanding how is it that evolution is able, with this relatively both powerful and so it's a powerful tool because you're getting lots of mutations, but it's also a very weak tool because the combinatorics is way, way, way more than the number of options that you're exploring. But with all of those things that we just pointed out and some other mechanisms, together evolution could achieve what it achieves. And as you can see, there's also some hand waving here. You need to go more deeply into it, and actually there's some things we may not even fully understand on how they're, we need to quantify them better and see how they're working in different environments. The mutation rate could also arise, become higher, for example, in bacteria under some stresses. So it's also super interesting, like of the situation when you need the mutations because you're under strong stress, maybe then the mutation rate becomes higher or not. It's still something that's being explored, etc.
What is the mutation rate of mutations which are not single nucleotide, like, I don't know, duplication, deletion?
Okay, so just before I answer that, that's another thing we didn't write, and that's why I wanted to end with this, because I think that there's so many different mechanisms so now that it makes you now wonder and explore about. So it doesn't have to be single base mutations. You could have other mechanisms happening with evolution playing around with mutations. What kind?
Deletion, insertion.
So it could be all sorts of rearrangements of the genome. What else? Okay, there's indels. Indels is like insertions, like you could have something getting inside and changing it, or you can have deletions, like deleting parts.
Duplications.
You could have duplications of the whole gene, which is then great because now you both have higher level, but then now you can make mutations that even if it's something that's supposed to be conserved, you can still conserve one copy, and the other copy could evolve, and you can play around even on conserved things. Other stuff?
Splicing.
What?
Splicing.
Splicing. So this thing with the splicing that seemed strange, why is it tenfold more of those introns and the exons and all that? You can also play around with how these things are getting combined and connected to each other, and that also gives you much more variability. It could be kind of like, it wouldn't be exactly these two mutations, but could achieve functional results might be similar. You could also take a whole chromosome and replicate it. You could have double of the chromosome, then many things go up together, and then you could play around with them. So all of those extra mechanisms are also at play. Now you were asking about the rates. So that's like, as you can guess, it's a long answer to quantify. It's also context dependent, organism dependent, condition dependent. It seems like very often you would find that also these are like, qualitatively important mechanisms. Like that many times when you're sequencing, say we're doing lab evolution, the group of Tzachi Pilpel here is a world expert in doing lab evolution, many other groups around the world, and you find that very often when you're posing a challenge, it wouldn't necessarily be a base pair mutation. It would be something like recombination, chromosome duplication, indel. That's like a large fraction of the cases. But also single base, but that's not a negligible mechanism. But there's several mechanisms that evolution is playing around with. So the fact that evolution has these several mechanisms, and it has all sorts of these other things, like recombining in sex, and selective advantage for even just one mutation that becomes even stronger when there's more. All of these things come together. Another example is like a random drift. Okay, you also have the fact that it's not that like all of us have completely the same genetic content. Even from the beginning, let's say if something even doesn't have an advantage, you're getting these mutations, and they're accumulating and accumulating and accumulating. And then it might turn out that now you need a new mutation, like 2 mutations. You just need to have the extra one. The other one you already have from before and it didn't do anything for you. But you just had a large natural variation in the population. And this is based on drift, the fact that the genomes, just drift without with being neutral. It's called neutral drift. So that's another mechanism that comes into play. Okay, so I want to leave you with this challenge of evolution, which I think is like at the cutting edge of trying to understand life in quantitative terms. You see there's different, it's both gives us like really good paradoxes to try and understand how it could do things, and different mechanisms in order to enable that. Any questions, comments, thoughts about this?
So we talked about different humans, but what about the cells within our body? Because when we multiply, the amount of humans that we can also, because we all started from like one cell, right?
Yeah, so it's true that also within our body, different cells would collect a different number of mutations. By the way, we could see, so, you know, given the number of cells, we could see how many replications has went past. And actually, you'll find that, like, that, you know, it's kind of like the calculation that we did here, number of replications, genome length, so you have a few mutations in each cell of our body. In terms of selective pressure, it doesn't usually help, like if I need to deal with Ebola, maybe, you know, one cell in my body has a mutation that's relevant to that, that wouldn't enable me to survive. So I really need the mutation to reach the zygote, and for my zygote to reach my daughters. And that would mean that now my daughters would be resistant because they'll be carrying the mutation throughout their body, and the fact that I have it just in my zygote wouldn't help. So it's kind of like, you know, the mutation arises, let's say, in me, but it's actually full-fledged in its effect just a generation later. Good. So with that, I'll thank you for, you know, participating throughout the class just as nicely as you did today. We'll be meeting for your final projects. If you have, you know, comments, thoughts, also feel free to be bold, you know, try new things. I hope that some of it would also be useful for you, either for your research right now or for things you'll be doing afterwards in life. I enjoyed giving the course and all this interaction. And if you like it, you can pass it on to your friends. It's all available, you know, on the web, and maybe we'll also have it next year. Toda.